Cells of higher organisms use cell organelles to separate metabolic processes from each other. This is how cell respiration takes place in the mitochondria, the cell's power plants. They can be compared to sealed laboratory rooms in the large factory of the cell. A research team at Goethe University has now succeeded in creating artificial cell organelles and using them for their own devised biochemical reactions. Biotechnologists have been attempting to reprogram natural cell organelles for other processes for some time, with mixed results, since the laboratory equipment is specialized on the function of organelles. Dr. Joanna Tripp, early career researcher at the Institute for Molecular Biosciences, has now developed a new method to produce artificial organelles in living yeast cells. To this end, she used the ramified system of tubes and bubbles in the endoplasmic reticulum, that surrounds the nucleus. Cells continually tie off bubbles, or vesicles, from this membrane system in order to transport substances to the cell membrane. In plants, these vesicles may also be used for the storage of proteins in seeds. These storage proteins are equipped with an address label, the Zera sequence, which guides them to the and which ensures that storage proteins are packaged there in the vesicle. Joanna Tripp has now used the address label, Zera, to produce targeted vesicles in yeast cells and introduce several enzymes of a biochemical metabolic pathway. This represents a milestone from a biotechnical perspective. Yeast cells, the pets of synthetic biology, not only produce numerous useful natural substances, but can also be genetically changed to produce industrially interesting molecules on a grand scale, such as biofuels or anti-malaria medicine. In addition to the desired products, however, undesirable byproducts or toxic intermediates often occur as well. Furthermore, the product can be lost due to leaks in the cell, or reactions can be too slow. Synthetic cell organelles offer remedies, with only the desired enzymes with address labels encountering each other, so that they work together more effectively without disrupting the rest of the cell, or being disrupted themselves. We use the Zera sequence to introduce a three-stage, synthetic metabolic pathway into vesicles, Joanna Tripp explains. We have thus created a reaction space containing exactly what we want. We were able to demonstrate that the metabolic pathway in the vesicles functions in isolation to the rest of the cell. The biotechnologists selected an industrially relevant molecule for this process, mucanic acid, which is further processed industrially to adipic acid. This is an intermediate for nylon and other synthetic materials. Mucanic acid is currently won from raw oil. A future large-scale production using yeast cells would be significantly more environment-friendly and sustainable. Although a portion of the intermediate protocataquic acid is lost because the vesicle membrane is porous, Joanna Tripp views this as a solvable problem. Professor Eckhard Bowles, head of the Department of Physiology and Genetics of Lower Eukaryotes, observes, "This is a revolutionary new method of synthetic biology." With the novel artificial organelles, we now have the option of generating various processes in the cell anew, or to optimize them. The method is not limited to yeast cells, but can be utilized for eukaryotic cells in general. It can also be applied to other issues, e.g., for reactions that have previously not been able to take place in living cells because they may require enzymes that would disrupt the cell metabolic process. Story source: materials provided by Goethe University Frankfurt. Note: content may be edited for style and length. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.